So we're going to be looking at the irreversible reactions of glycolysis. These are the reactions catalyzed by the following enzymes: hexokinase, PFK1, and PK. So for PFK is phosphofructokinase 1, PK is pyruvate kinase. So the first reaction is hexokinase, where glucose is converted to glucose 6-phosphate and ATP is being used. The other reaction is the one for PFK1, which is reaction 3 of glycolysis, and fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and also ATP is being used. And then the other reaction is reaction 10 of glycolysis, which is the final reaction of glycolysis, where pyruvate kinase is the enzyme, and phosphoenol pyruvate is being converted to pyruvate, and phosphoenol pyruvate is a high energy compound, and that's why here we're going to have the substrate level phosphorylation. If you can remember in the previous video, and here ATP is being formed. The next question will be the reversible reaction of glycolysis, which involves ATP. So this is going to be reaction 7 of glycolysis, where we're going to have phosphoglycerate kinase as the enzyme, and 1,3-bisphosphoglycerate is going to be converted to 3-phosphoglycerate, and this 1,3-bisphosphoglycerate is a high energy compound. So of course here we're going to have substrate level phosphorylation, where ATP is going to be formed. Our next question would be, what are isoenzymes, and an example. So isoenzymes are enzymes that catalyze the same reaction but have different enzyme kinetics, the Km. For example, the enzyme kinetics can be Km. So an example would be hexokinases, and hexokinases are of different types. Like we're talking about isoenzymes, we have type 1 to type 4, and type 4 is a bit special. It's also known as glucokinase or type D, and it's found in hepatic cells, for example the liver and the pancreas. Yes, so that's all about isoenzymes. The next question would be, what are the allosteric regulation of enzymes of glycolysis? How are enzymes allosterically regulated in glycolysis, meaning both inhibition and activation, increasing activity? So the first one will be hexokinase, which we've just seen is an isoenzyme. From type 1 to type 3 though, type 4 is not allosterically regulated. So type 1 to type 3, they are inhibited by accumulation of the product. What is the product? Glucose 6-phosphate. And the reaction we're talking about here is the first one for hexokinase, where glucose is converted to glucose 6-phosphate. So for hexokinase, they are inhibited by accumulation of the product. The other enzyme that is allosterically regulated is PFK1. Just to remember the equation, we're talking about equation 3 of glycolysis. We're talking about equation 3 of glycolysis, whereby fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and ATP is being used. So it is allosterically regulated. It is its activity is increased by ADP, AMP, and fructose 2,6-bisphosphate, which is an important allosteric regulator. So ADP, AMP, and fructose 2,6-bisphosphate, and it is inhibited by increased ATP, whereby ATP is both a substrate and an inhibitor. And the last enzyme that is allosterically regulated in glycolysis is PK. And just to remember the equation, it's this equation 10. Equation 10, where phosphoenol pyruvate is converted to pyruvate, and ATP is formed because phosphoenol pyruvate, or what we've written as PEP, is a high energy compound. And so it's being inhibited by ATP and acetyl-CoA, well, it's being activated by fructose 1,6-bisphosphate. So our next question would then be what is the rate limiting step and enzyme of glycolysis? The enzyme will be PFK1, and the reaction is reaction 3 of glycolysis, where fructose 6-phosphate is being converted to fructose 1,6-bisphosphate using the enzyme PFK1, and ATP is being used. And then what is an important regulatory site in glycolysis? So the enzyme is PK, pyruvate kinase, and the reaction is reaction 10, which is the last reaction of glycolysis, where phosphoenol pyruvate, a high energy compound, is being converted to pyruvate, catalyzed by pyruvate kinase, and ATP is being formed at substrate level phosphorylation. So that's the important regulatory site. The next question would be a reaction in glycolysis with a large negative standard free energy, and this would still be reaction 10, and where phosphoenol pyruvate is being converted to pyruvate, ATP is being formed, PK is the enzyme catalyzing the reaction, and this large standard free energy is negative 34.4 kilojoules per mole. The last question we have here for glycolysis would be which molecule is involved in regulating hemoglobin's affinity for oxygen? Which molecule in glycolysis is involved in regulating hemoglobin's affinity for oxygen? This would be 2,3-bisphosphoglycerate, and if you're wondering how we got 2,3-bisphosphoglycerate from glycolysis, this is the reaction. Before we discuss the reaction, we can talk about 1,3-bisphosphoglycerate, which is a reaction number 7. The normal reaction of 1,3-bisphosphoglycerate involves it being converted to 3-phosphoglycerate, catalyzed by phosphoglycerate kinase, and it's a substrate level phosphorylation where ATP is going to be formed. However, here we want 2,3-bisphosphoglycerate, and we're going to form it from 1,3-bisphosphoglycerate catalyzed by bisphosphoglycerate mutates to form 2,3-bisphosphoglycerate. This 2,3-bisphosphoglycerate can still be converted to the normal product 3-phosphoglycerate catalyzed by 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate phosphatase. However, we're interested with this first part of the reaction where 1,3-bisphosphoglycerate is being converted to 2,3-bisphosphoglycerate catalyzed by bisphosphoglycerate mutase. So that's all we had for this video and about glycolysis. Our next video we'll be looking at the TCA cycle. Thanks for watching.